I think while people are joining, can you hear me? I think we should slowly start so that we don't delay our today's Cardiovascular Research Discoveries series seminar that the Journal of Cardiovascular Research, the Basic and Translational Sciences Journal of the ESC, is co-organizing together with Council for Basic and Cardiovascular Sciences, led at the moment by Christian Weber, who has kindly joined us. Christian will introduce the first speaker. Today's topic is cardiovascular fibrosis, and we have three speakers who have through their recent work and sometimes lifetime work, usually contributed to this field. We are very grateful for them joining us, and I would like to ask Christian to introduce our first speaker.
Yeah, thanks very much, Tom, for opening, and I'm delighted to introduce Nick Frangogiannis, who is a pioneer in the field, actually, that we're going to discuss today. He is a professor of medicine and a professor at the Department of Microbiology and Immunology at Albert Einstein Medical College in New York, and an Edmund Zafra Chair of Cardiovascular Medicine. And as I've mentioned, Nick has pioneered the role of myocardial inflammation and cardiac fibrosis in the context of myocardial infarction and heart failure, in particular the role of specific cell subsets such as macrophages, fibroblasts, and parasites, and also the role of the molecular determinants. And this is where basically our paths also crossed as pertaining to the function of chemokines in these processes, but also he's looking at the role of TGF-beta and matricellular proteins. And in the course of that, he's published more than 200 seminal publications in that field, and accrued an H index of 79. So really a highly significant figure in the field, and we're looking very much forward to your presentation.
Thank you very much, Christian. Thank you, Tom. Thank you for inviting me to speak in this very interesting series. I will start by sharing my screen. I hope everybody can see it. So I will discuss concepts related to cardiac fibrosis. And this is a vast topic. So what I will be talking about after providing some definitions is some basic concepts on the cell biology of cardiac fibrosis, focusing on fibroblasts, myofibroblasts, and a cell that we have been particularly interested in recent years, the pericyte. I will discuss some interesting directions on the diverse functions of the fibroblasts in myocardial disease, and I will close with some therapeutic perspectives. So what is cardiac fibrosis? Cardiac fibrosis is defined as the expansion of the interstitium through enhanced deposition of extracellular matrix proteins. It's important to realize that cardiac fibrosis is not a single disease. It's not even a group of diseases, but it's a response which accompanies most myocardial pathologic conditions. And it is a response to injury that can be reparative or maladaptive. And sometimes even the crude pathologic analysis gives us some information on whether this is reparative or potentially maladaptive when we have replacement fibrosis, such as in myocardial infarction. In the non-regenerative organ, that is the heart, we have replacement of dead cardiomyocytes with scar tissue, and this is clearly reparative. In other cases, when there is no loss of cardiomyocytes, where we have interstitial or perivascular fibrosis, this process can be maladaptive, although it could have reparative components as well. And several cell types have been implicated in the pathogenesis of fibrosis, including immune cells, vascular cells, cardiomyocytes, platelets. All these cells can contribute by producing cytokines, growth factors, and matricellular proteins that will then activate the fibroblasts, or by having effects on the matrix, by affecting matrix remodeling through the production of proteases and antiproteases. However, there is only one cell type, the fibroblast, which can produce structural extracellular matrix proteins. And this is the reason why fibroblasts and their activated form myofibroblasts have such a central place in the pathogenesis of fibrosis. It's essential in science to ask important questions. And this is a famous painting by Paul Gauguin, which was painted in Tahiti and it's currently in Boston. Its title is D'où venons-nous, que sommes-nous, où allons-nous? In English, where do we come from? What are we? Where are we going? These existential questions are particularly important in understanding fibroblast biology. So we could ask similar questions on the fibroblasts. What are the fibroblasts? Where do they come from? What is their purpose and function? And where do they go? What's their fate after they have served this role? So in the next few minutes, I will try to answer some of these questions, starting with the most fundamental one. What are the fibroblasts? Fibroblasts have been described by Virchow in the 19th century, and little has changed in our ability to define them over the last 150 years. So we recognize fibroblasts as cells of mesenchymal origin that reside in connective tissues, lack a basement membrane, and secrete extracellular matrix proteins. So this is a morphological definition with a functional component as well. And unfortunately, there are no specific markers for fibroblasts. This is a table summarizing our knowledge on the sensitivity and specificity of various markers. And whenever you have such a long list of potential markers with so many references and information listed here, you know that there is not a single marker that you can use to identify the fibroblasts. What one needs and what we often use for a systematic identification and characterization of fibroblasts is a combination of relatively non-specific markers and some exclusion criteria. And this is a very nice figure from one of the papers by Sylvia Evans doing exactly that. So they're using a collagen reporter mouse. Fibroblasts are collagen producing cells, relatively non-specific, but a pretty good marker. And then a relatively specific marker in PDGF receptor alpha, which identifies fibroblasts. Vimentin, which is not particularly specific because it labels all mesenchymal cells, and then what these cells are not. So they're PKAM1 negative, they're non-endothelial cells, they're CD5 negative, they're non-hematopoietic cells, and then PDGF receptor beta, which is expressed by mural cells and by subset of fibroblasts, is expressed in just as a population of fibroblasts, and alpha-spassalactin is a marker of activated myofibroblasts, but not of quiescent fibroblasts. Fibroblasts have two major characteristics, heterogeneity and phenotypic plasticity. And we came to appreciate heterogeneity of fibroblasts more and more with the advantages that single cell work has given us. And some of the best work, some of the best single cell transcriptomic analysis of cardiac interstitial cells came from Australia. One seminal study is the one that you see here by Alex Pinto, who is going to discuss, I'm sure, these concepts much more in his presentation. And the other is by Richard Harvey in eLife, identifying clusters of fibroblasts with distinct transcriptomic profiles, a palette of colors in where the fibroblasts are identified, and suggesting that each one of these clusters may have distinct functional profiles. The other major characteristic of fibroblasts is their phenotypic plasticity. So they respond to their environment with dramatic transformations. And an example is in vitro. If you culture fibroblasts in plates, then they become myofibroblasts. They express alpha-spassalactin. You can see here the stress fibers, which are decorated with alpha-spassalactin. But if you take these same cells and you put them in an environment where they're in contact with collagen and they're in a free-floating pad, so they're shielded from mechanical stress, then these same cells disassemble their stress fibers and they have very low expression of alpha-lactin. They exhibit a dendritic morphology, which is more closer to the phenotype that quiescent fibroblasts have in tissues. And of course, in vivo, these transitions are even more dramatic, and they can have many different forms of fibroblast phenotypes depending on context-dependent factors, on secreted mediators, and on mechanical stress. In most fibrotic conditions, there is an expansion of the fibroblast population, and this is typical in infarction. This is from our old work in canine infarcts, showing the well-aligned arrays of activated myofibroblasts in the infarct border zone. So these cells expand. However, where do they come from? Where does this increased population of fibroblasts comes from? And when I joined Mark Entman's lab back in the 90s as an internal medicine resident at that time, the answer to this seemed to me quite simple. So these are some pictures that I took myself with a fluorescent microscope, an old traditional fluorescent microscope with a camera hooked on top of it, back in the days where we would stick adhesive labels as A and B rather than using digital technology. And you can see here that these myofibroblasts, which are stained with alpha-lactin, are also positive for the proliferating cell nuclear antigen, this red staining here. So it's a combination of light and fluorescence. So my answer to this question would have been at the time that there are enough fibroblasts in the heart, the environment is conducive to proliferation, so their expansion comes from proliferation of local populations. Why would you need anything else? Why would you need another cell type to contribute? However, in the next few decades, there was experimental work from several labs suggesting that other cell types can transition to fibroblasts, thus building this concept of plasticity, of cellular plasticity, that endothelial cells can become fibroblasts, that bone marrow-derived cells can become fibroblasts, macrophages can become fibroblasts. Practically every cell type could contribute to the fibroblast population. Recent work and more robust work using specific drivers and actually reliable methods for identification of fibroblasts have suggested that these sources are not major sources of fibroblast populations in infarcted and remodeling hearts. And this is work from Jeff Molkentin, Sylvia Evans, Reza Ardehali that suggested this. However, there is one aspect of the question on the origin of fibroblasts that remains unexplored, and this is the potential role of the pericytes, the mural cells, which are quite abundant in the heart. And we are particularly interested in the pericytes for two reasons. First, because there is a large number of pericytes in the heart, and this is the atlas of the heart that Sarah Teichmann published, showing a very large population of mural cells in the human heart, which actually outnumbers the fibroblasts. I have to admit that this is not necessarily confirmed with other single cell protocols, but definitely there is a large population of mural cells in the myocardium. And then in other organs and in other systems, there is extensive work suggesting that these cells are plastic and they can convert to fibroblasts. So this is one of the papers by Raphael Kraman showing that in several different tissues, GLI1 positive perivascular cells contribute to the fibroblast population. Of course, what is unclear is whether these GLI1 positive cells are indeed mature pericytes or perhaps some adventitial fibroblasts. To address this question, we first validated our tools. And after looking at the testing several options, we found that the most reliable tools for identification, labeling, and tracing of fibroblasts is the NG2 reagents, the NG2 mice, NG2 reporter mice, and the inducible NG2 mice. And we use the PDGF receptor alpha as a tool to label fibroblasts concurrently so that we can study the conversion of the fibroblasts with inducible pre-driver into myofibroblasts, the conversion of the pericytes into fibroblasts. And we used this tool, these inducible NG2-CRE mice with the PDGF receptor alpha reporter in a model of myocardial infarction. And we found that a very small fraction of infarct fibroblasts is derived from pericytes, was about 4%. You can see all these green nuclei of fibroblasts, which are for the most part not derived from pericytes. The number of myofibroblasts, the way that we defined myofibroblasts, was higher. So it was about 15% of the myofibroblasts that were derived from pericytes. Importantly, however, when we sorted these pericyte-derived fibroblasts, they were overactive. So their transcriptomic profile showed high levels of expression of collagens, MMPs, TIMPs, matricellular genes, high levels of integrins and MMP14, suggesting perhaps a migratory phenotype. So they were overactive in comparison to fibroblasts from other sources. So perhaps they could contribute disproportionately to fibrosis because of this overactivity that they show. What is certain about pericytes, about cardiac pericytes in the heart, is that they contribute to infarct angiogenesis and actually to regulation of infarct angiogenesis. In the infarcted heart, you have formation of abundant immature vessels which have a poor pericyte coverage, and then during the proliferative phase there is recruitment of these neural cells into these vessels. You can see here these red cells, these clusters of red cells, as they accumulate into these forming vessels. They almost occupy the entire pseudolumen here, and then eventually at the 28-day time point they form a vessel with a thick media, which I doubt can serve as a conduit of blood, but most likely this process serves to stabilize the vasculature, to prevent inflammation, and also to inhibit angiogenesis. We looked at what are the mechanisms for these pericyte transitions, and we identified the TGF-beta pathway actually as a candidate for the fibrogenic conversion of the pericytes. So we generated mice that lack the type 2 TGF-beta receptor, specifically in pericytes, and these mice, in response to myocardial infarction, to our surprise, had no difference in fibrosis and in myofibroblast density. However, they were defective in terms of formation of these mature vessels. There was a reduced density of mature vessels in these animals. So to summarize this and published work that was done in the lab by Linda Alex and now is continued by Isabella Tuleta, in the infarcted heart, a subset of pericytes converts to fibroblasts and accounts for about 10 to 15% of infarct myofibroblasts and much fewer percentage of the overall fibroblast population. The pericyte-derived fibroblasts are overactive, expressing large amounts of extracellular matrix genes, and the pericytes contribute to infarct angiogenesis by coating infarct neovessels. So the majority of infarct fibroblasts and myofibroblast are derived from resident fibroblasts. So the question is, what is their function? What do they do? The fibroblasts have a diverse range of functions in different contexts, and even in the same disease, as the environment changes, fibroblasts appear to play different roles. So fibroblasts have been suggested to serve as pro-inflammatory cells. They can activate the inflammasome, they can produce cytokines and chemokines. Are they an important source of cytokines and chemokines in the healing infarct? It's unclear because several other cell types, including immune cells, macrophages in particular, endothelial cells, can produce large amounts of pro-inflammatory cytokines. So the specific contribution of fibroblasts remains elusive. What is certain is that fibroblasts serve as reparative cells. They form these arrays in the infarcted heart, they deposit the matrix, and they protect the heart from rupture, and they protect the heart from adverse dilative remodeling by increasing the tensile strength of the scar. And this is suggested by many studies that disrupted in a fibroblast-specific manner pathways critical for activation of a reparative phenotype in the fibroblasts, and found defects. And this is from our own work looking at the effects of myofibroblast-specific SMAD3 loss, SMAD3 being one of the key downstream signals of TGF-beta signaling, showing the development, the increased mortality in these mice, the development of rupture, and also development of dilation, adverse dilative remodeling. And this was due to formation of a chaotic scar, as the fibroblasts in this scar were misaligned, thus producing and generating reduced tractional forces. And also there was disruption of the extracellular matrix network, and this was related to effects of SMAD3 on an integrin reactive oxygen species axis. Fibroblasts also play protective roles in models of non-reparative fibrosis. That came to our surprise when we did experiments using the myofibroblast-specific SMAD3 knockouts in a model of left ventricular pressure overload, in which we found that fibroblasts are important to preserve the matrix around cardiomyocytes under conditions of stress. So this is a published paper, so I'm not going to show the data, but this cartoon, what it shows, it illustrates the events occurring in the pressure overloaded heart, induction of reactive oxygen stress, inflammatory pathways, neurohumoral activation. All these result in matrix damage and generation of matrix fragments. And the fibroblasts responding to TGF-beta SMAD signaling, they preserve the integrity of the matrix by reducing collagenase expression and by increasing TIMP1 expression. And this is very important for the cardiomyocytes, which require a matrix network around them to survive and to function properly. And also it's important to prevent formation of matrix fragments, which would have pro-inflammatory effects, so to attenuate inflammation. This is not the only function that fibroblasts can have in relation to the cardiomyocytes. There are findings from other groups. Thomas Toom's group showed that they produce exosomes that contain miRNAs that can have effects on cardiomyocyte hypertrophy. Ryozo Nagai's group have suggested they can produce paracrine mediators such as IGF-1, and they can actually mediate adaptive responses important to the pressure overload. So they are not just matrix producing cells, but actually they release signals important for homeostasis and important for the response of the cardiomyocytes under conditions of stress. So all these sound protective. So why do they have a bad reputation? So is there anything detrimental that the fibroblasts do? And the answer is yes. When they are uncontrolled, when they are unrestrained, when the endogenous mechanisms responsible for their quiescence after they have produced large amounts of matrix fail. And we found that through our work on the inhibitory SMAD7. SMAD7 is one of the two inhibitory SMADs that is known to serve as a negative regulator of the TGF-beta cascade. And Claudio Humeres did the work on this that was recently published. And he found that myofibroblast-specific loss of SMAD7 is associated with fibrosis after myocardial infarction, which results in dysfunction and actually in increased mortality, which is not related to rupture, but is associated with heart failure. And he then identified the mechanism through which SMAD7 acts. And he looked at what level of the TGF-beta cascade SMAD7 acts. This is comparison of SMAD7 null cells versus the control cells in terms of their response to TGF-beta for activation of the TGF-beta receptors, the receptor-activated SMADs, and non-SMAD pathways. And he found that the effects of SMAD7 are downstream of the receptors, which are not affected by the absence of SMAD7. But as you can see, phospho-SMAD2 and phospho-SMAD3 are markedly increased in the absence of SMAD7, as well as some of the non-SMAD pathways, such as ERK and the AKT. Importantly, Claudio discovered a TGF-beta independent effect of SMAD7. This came from unbiased work looking at our RNA-seq data and identifying receptor tyrosine kinases as differentially regulated by SMAD7 absence. Then he did a protein array where he identified several candidates of these receptor tyrosine kinases. And finally, this is the end stage of this quest. He found that in the absence of SMAD7, there is overactivity of ERBB2 in the fibroblasts, suggesting that SMAD7 inhibits the ERBB2 response. And he demonstrated an interaction between SMAD7 and ERBB2 in fibroblasts. And he found that this interaction is responsible for part of the effects of SMAD7, and in fact, in a TGF-beta independent manner. To summarize our findings on the fibroblasts, fibroblasts have a wide range of functions in diseased hearts. They may activate inflammatory pathways, they may deposit matrix, may regulate matrix remodeling, they may protect cardiomyocytes. There are other potential functions that I have not discussed. They may promote angiogenesis as well. Following myocardial injury, fibroblast activation is important in repair. However, unrestrained or excessive fibroblast activation contributes to the pathogenesis of heart failure. And let's see why. Okay. So a key question here is whether this functional diversity of fibroblasts is reflective of their heterogeneity. So do the different transcriptomic profiles of the fibroblasts correspond to distinct functions? And how do we integrate heterogeneity and plasticity of the fibroblasts? So these are two different concepts, one suggesting that the different functions of the fibroblasts are due to the presence of different subsets, and the other is that there is a continuous transition of the entire fibroblast population. We need somehow to reconcile these two, to integrate these two, and to examine how different subsets may transition through the phases of injury and into repair. And importantly, which signals mediate this phenotypic transition. So these are all important questions for our basic research for understanding the biology of fibroblasts. However, we're not doing that just out of curiosity, just to learn what the fibroblasts may do in any relevant model of injury. We do it because we feel that this has a chance of contributing to the patient's well-being, that interventions targeting cardiac fibrosis may have some beneficial effects in patients with heart failure. This is an expert commentary by two of the best in the field, Javier Diaz and Rudolf de Boer in cardiovascular research, making a very strong statement on this. Management of cardiac fibrosis is the largest unmet medical need in heart failure. Which is actually true if one includes in there all these patients with replacement fibrosis, with infarctive ischemic heart failure, for which, however, there is not much that we can do in the absence of a regenerative approach, because we cannot replace an existing scar with new cardiomyocytes. So our options, and if this has applicability, is limited to patients with chronic heart failure who have perhaps interstitial and perivascular fibrosis. So how about antifibrotic therapies in this patient population? And I will offer some thoughts on this. First of all, cardiac fibrosis is not the primary disease entity. It's not like idiopathic pulmonary fibrosis or systemic sclerosis, in which you have a primary fibrotic disease that you know is causing the dysfunction, the organ dysfunction. It often reflects a reparative response even when you don't have significant cardiomyocyte death. And although in heart failure patients, the extent of cardiac fibrosis often is associated with adverse prognostic implications, this may not necessarily mean that the fibrotic remodeling has detrimental effects, but it may be an epiphenomenon and may reflect the severity of the primary injury. So at this point, we have very limited information on the clinical context. And there are some key questions to address here. So in heart failure patients, what is the contribution of fibrotic remodeling to systolic and diastolic dysfunction and to arrhythmogenesis? And perhaps can we identify patient subsets with maladaptive fibrosis? Certain patients may exhibit dysregulated or excessive reparative fibrotic responses that may adversely affect outcome. And although causation is impossible in the clinic, perhaps we can infer from our associative data a relation that could be targeted in this subset of patients. So what we need in the field is work for a pathophysiologically driven stratification in myocardial disease. We define currently heart failure the same way practically that was defined decades ago as a syndrome. And the attempts to identify subsets are based on superficial endpoints, very useful and very well integrated into the clinical practice, but not related to pathophysiology. So can we identify heart failure patients with prominent fibrosis that may be driving their dysfunction? And this could be done through a combination of strategies. Clinically defined groups, perhaps diabetic patients may have more fibrosis. This will be crude and would have implication in a small subset of the patients with heart failure. Circulating biomarkers may be a bit better, but again, they do not reflect just the myocardial response, and they have not been at this stage particularly useful in identifying such subsets. The promise is in imaging, and in particular in CMR, in terms of identifying patients with significant fibrosis and with progression of fibrosis, and perhaps even better to identify patients with specific types of changes in their interstitium that may benefit from specific antifibrotic strategies. And with these thoughts, I will close the presentation. Would like to thank the members of my lab, in particular, Claudio Humeres, who did all the work on SMAD7, and Linda Alex, who did the work on the pericytes, which I presented here. And thank you very much for your attention.
Thank you very much, Nick. This was really excellent overview of your own work, but in the context of major discoveries in sort of fibroblast biology and fibrosis. And thank you for also showing the real unmet needs in the field. And we will have a chance to discuss after the two short talks. The second presentation today will be delivered by Professor Alex Pinto, who is an associate professor and head of Cardiac Cellular Systems Laboratory based at the Baker Heart and Diabetes Institute in Melbourne, in Australia. So Alex is joining us in the middle of the night. We appreciate this very much. His research aims to understand the network of cells, and Nick has already referred to some of his important work published recently. He's using to address this cutting-edge multi-omics technologies and other imaging approaches that I hope we will hear from him about. Alex, please. Thank you for joining us.
Okay, thank you. I am unsure if I'm sharing my screen.
Yes, you are.
Okay, great. So thank you, Tom and Christian, for this invitation, and good morning, good afternoon, and good evening, everyone, depending where you're from. So as Tom noted, my lab is interested in understanding the different cell types of the heart, and particularly understanding fibrosis. But unlike some of the aspects Nick just discussed, I'm not as interested in replacement fibrosis, more interstitial fibrosis. As you can see here in this mouse heart cross-section, and I'm interested in how these interstitial fibrosis and fibroblasts develop and contribute to cardiac dysfunction. So I think we're at a stage where we're really getting some really interesting insights to the cellular and molecular drivers of fibrosis, and a key contributor to these new insights have been single cell omics. And today I'm going to share with you some insights I've gained from studying 2 models of fibrosis using single cell omics. The first of these is the angiotensin 2 infusion model. And in this model, we infuse male and female mice with Ang2, and we can induce hypertrophy and fibrosis, as you can see here. But like Nick showed in our paper, we're also able to do single cell transcriptomics. And not only are we able to map the cardiac cellular landscape in high resolution, but a feature of this particular study was the emergence of 2 activated fibroblast species, which we call fibroblast SILP and fibroblast THBS4, which emerge in context of this physiological stress. Now, regarding the contribution of different cells to fibrosis and extracellular matrix, we can consider every cell type in the cardiac cellome, this in the mouse, including cardiomyocytes, and examine cells, genes that are upregulated, shown here in orange, or those that are downregulated in gray. And when we consider the genes that are upregulated for every cell population and the pathways, these are the top 3 pathways that are upregulated for every cell population, we find that most cells, virtually all cells, upregulate genes involved in ECM and ECM remodeling. So what about fibroblast SILP and THBS4? When we compare these cells to fibroblasts from other Ang2 mouse hearts, we find that the genes upregulated in these fibroblast subsets represent those involved in ECM, again, wound healing and collagen deposition. And when we consider their cell number and the transcripts within them, in fact, we find fibroblast SILP and THBS4 are indeed the most fibrogenic cell types in this model of fibrosis and in our data set. Now, importantly, these aren't mouse-only phenomena. We can also show evidence of these cells in the human context, and we can do this by at least 2 means. Firstly, when we analyze bulk RNA-seq data from human hearts, each one of these dots represents an individual human heart, and divide these human hearts based on hypertrophic and non-hypertrophic, and then consider expression levels of relatively cell-specific genes. Now, these are genes that are enriched in fibroblast SILP and THBS4. We find these genes are upregulated in the hypertrophic hearts. And further, we can also, using our single-cell omic data, find markers of these cell populations. And here we've got failing human hearts, and we can see evidence of fibroblast THBS4 in this context. Importantly, we've been also been able to map the distribution of these cells within the heart, and we had some interesting findings here also. Firstly, that the THBS4 fibroblasts primarily aggregate in these focal fibrosis regions, particularly around the hinge points. Whereas in the perivascular regions, the THBS4 fibroblasts are almost completely absent. So this is important because it suggests perhaps the evolution and the development of these cells might have some sort of spatial origin or spatial contribution towards them. Now, when examining these cells and looking, trying to seek an identity for them or assign an identity for them, we looked at a range of different activated fibroblast markers. Some of them are summarized here. When we statistically compared genes that are highly enriched for these fibroblast SILP and THBS4, we found SILP and THBS4 are the most enriched. And then others were, some were enriched well in these cells and others less so. But one of the most striking findings here was that the ACTA2, a gene that codes for smooth muscle actin, or transcripts for it, weren't enriched in fibroblast SILP and THBS4. Whereas you can see it, this transcript's high in smooth muscle cells. We can also show that these cells aren't myofibroblasts by histological staining. Here we've stained for these fibroblasts and these fibrosis and THBS4 fibroblasts, and we can also stain for ACTA2. And really these experiments are internally controlled because we can see arterioles corresponding to ACTA2 positive cells within this tissue, but these fibrosis regions are not ACTA2 positive. Now that's really important, I think, because a lot of our research is trying to understand fibrosis, using ACTA2 and myofibroblasts as a model of primary driver of fibrosis. And the fact that we don't see myofibroblasts in this type of fibrosis model, despite seeing extensive diastolic dysfunction, I think is important for instructing us about how we look at mechanisms driving fibrosis and therapeutic targets for ameliorating fibrosis. So the fact that these cells aren't myofibroblasts isn't due to lack of TGF-beta1 expression. Here we're showing the atlas once again with TGF-beta1, which is upregulated in many different cell types in the heart. But one reason we think perhaps they're not developing into myofibroblasts are because of transcription factors and matricellular proteins, and also other paracrine factors, which all regulate TGF-beta1 activity. So we think these different pathways are reasons why myofibroblasts aren't perhaps forming in this model. Now, we also looked at inflammation and whether inflammation is important for the development of fibrosis. Now, indeed, when we did flow cytometry, we could find inflammation in this model, but we could only see evidence of inflammation looking at myeloid cell infiltration to the heart in the male context. Now, I think this is also important because in the females, we see extensive fibrosis, we see diastolic dysfunction again, but no inflammation. And this really instructs us, I think, or gives us a new perspective to consider the role of inflammation in driving fibrosis and particularly diastolic dysfunction. In fact, we could see inflammatory pathways enriched in one context in our data set, and that was amongst the ligand genes that are downregulated. And when we look at the pathways those genes represent, those included those involved in cell chemotaxis and inflammation. So we also wondered in a different study, does inflammation yield fibrosis? And to do that, we looked at high-fat diet, and this was a collaboration with Sean Bender from University of Missouri. And in these mice, these were female mice, after giving them high-fat diet, we did single-cell transcriptomics. And here also, we could see hundreds of genes up and down regulated in this model. But a feature here amongst the top upregulated pathways were those involved in inflammation. So in the high-fat diet, we see extensive inflammation, systemic inflammation, and also local inflammation. But fibrosis isn't a key feature of pathological remodeling of this heart. In fact, when we looked at collagen genes to just verify that collagen genes aren't upregulated in this context, we couldn't see upregulation of these genes. We couldn't see fibrosis in the interstitium or perivascular localization. In fact, we could see some fibrosis genes enriched in one context, and those were amongst the downregulated genes. So what this tells us is fibrosis isn't dependent on inflammation, nor does inflammation drive fibrosis necessarily. And I hope this single-cell omics data has given you a sense of how many different cell types that can contribute to fibrosis, and that's many. Although our findings were that fibroblast SILP and THBS4, two activated fibroblast subsets, are the primary drivers of fibrosis. So thank you, everyone. So this work, of course, is not possible without the contributions of many people and many collaborations, and I've listed them here. Thank you very much.
Yeah, hello. Can you hear me? Thank you
Yes,
very
we
much
can
for
hear
this
you.
nice presentation. And could you please unshare maybe? That would be very nice. And with this, I would like to introduce our last speaker, Ryszard Nosalski. Ryszard is a postdoc at the Institute of Cardiovascular and Medical Sciences at the University of Glasgow. And he has earned his PhD degree in medical science from the Jagiellonian University College of Medicine in Krakow. So basically following on Tom's path and guidance, and he has contributed to many projects and publications. One of them is a recent Nature paper that we had a shared contribution in that was also looking at the neuroimmune interfaces in atherosclerosis, and his major interest is indeed the immune response in cardiovascular disease and how inflammatory mechanisms can underlie the development of endothelial dysfunction and perivascular inflammation and also arterial fibrosis, also in the context of hypertension. So we look very much forward to your short presentation.
We don't hear.
Okay, so before I will start, I would like to thank you for this introduction and invitation and opportunity to present my study. So vascular fibrosis is the most characteristic feature of the vascular remodeling associated with hypertension. It's driven by the fragmentation and loss of elastic fibers and accumulation of stiffer collagen fibers in the vascular wall and vascular stiffening. So the main determinants of arterial stiffening is, and fibrosis belongs oxidative stress and perivascular inflammation, which is manifested by enhanced accumulation of the immune cells in perivascular adipose tissue. In the close proximity of adventitia, where the highest accumulation of the collagen is observed. This process can be regulated by epigenetic mechanisms such as non-coding RNA, which are involved in gene expression regulation. However, the role of microRNAs in the regulation and modulation of perivascular inflammation and fibrosis remains undefined. So we screen the microRNAs that are changed within the perivascular adipose tissue in hypertensive fibrotic vessels. ANG2 was used as a model and was associated with a clear separation of normotensive and hypertensive animals in the principal component analysis, compromising all MIR transcripts. Out of 381 screened microRNAs, 15 were upregulated and 13 were downregulated. However, MIR214 was the only overexpressed after Bonferroni correction with 8-fold increase. And surprisingly, the increase was not attributed to the primary adipocytes isolated from perivascular adipose tissue of these mice. So this is interesting because the MIR214 was shown to be important in the other organs and pathologies, including fibrosis. So we used the MIR214 knockout animals and we infused them with angiotensin, and histological evaluation of aortic samples showed a significant reduction of adventitial collagen in MIR214 knockout animals in comparison to wild type after picosilius red and Mason-Frichem staining. This was also observed in the aortic hydroxypyrroline level, which is a major component of the protein collagen protein, and as well mRNA expression for collagen 1, 3, and 5, which was upregulated only in wild type but not in knockout animals, while fibronectin and TGF-beta increased in wild type hypertensive animals. Only TGF-beta protein level was abolished in MIR214 knockout mice, indicating indirect regulation of TGF-beta in this model. So enhanced collagen deposition upon Ang2 infusion caused marked stiffening of the aorta in wild-type animals as evidence of left-forward shift of strain-stress relationship studied by pressure myography. In contrast, MIR214 knockout animals had significantly no effect on aortic stiffness. Next, we additionally performed nano-intentional studies utilizing atomic force microscopy, and which we also observed the increased Young modulus in wild-type hypertensive animals, but not in the knockout hypertensive animals, confirming the importance of MIR214 for the vascular stiffening in mouse model of hypertension. In order to provide the translational context, we studied the serum MIR214 level in the context of vascular stiffness as a hallmark of vascular fibrosis. And MIR214 level was significantly higher in hypertension and was significantly correlated with plasma MIR214 level and pulse wave velocity as a clinical measure of vascular stiffness. Furthermore, this correlation was observed only in hypertensive patients. So MIR214 level was upregulated also in peripheral blood and spleens. However, this induction was related only to CD3 positive cells in peripheral and T cells isolated directly from perivascular adipose tissue. And interestingly, MIR214 did not rise in perivascular adipose tissue of RAG1 knockout animals, which are lacking of mature T cells, despite of Ang2 infusion, which indicates the critical role of T cell in MIR214 induction observed in the perivascular adipose tissue. T cell activation requires binding of the T cell receptor with the antigen presenting on the membrane of antigen presenting cells. Experimentally, we can mimic this by exposition of cells to anti-CD3 antibodies, and interestingly, this was associated with upregulation of MIR214 in the T cells. Keeping this, we infused RAG1 knockout animals with Ang2, and they were protected against enhanced collagen accumulation around the vessels. To provide the functional role of T cell MIR214 in the fibrosis, we performed adoptive transfer of T cells collected from wild type and MIR214 knockout mice. After 14 days of infusion, we noticed similar level of circulating T cells in periphery. However, the perivascular fibrosis was restored only upon wild type T cell transfer, but not when the MIR214 deficient T cells were transferred. And this was associated with the reduced aortic collagen expression in RAG1 knockout animals transferred with MIR214 deficient T cells when compared to wild type T cells, which indicates the crucial role of T cell MIR214 in adventitial collagen accumulation. To understand how MIR214 in T cells could modulate the profibrotic action in hypertension, we perform RNA-seq analysis of isolated T cells, and we noticed that Ang2 in vivo induced change of 1,380 genes in wild-type animals, while MIR214 knockout T cells remained greatly unresponsive, with only 51 genes altered. Moreover, a large number of the top genes which were significantly downregulated in wild-type T cells, but not in the knockout T cells, were marked as a predicted targets for MIR214, indicating the pleiotropic role of microRNA in transcriptional reprogramming of T cells in Ang2 hypertension. Genes and enrichment analysis showed many pathways affected by Ang2 in wild type, but not in knockout T cells, mice including cytokines release and T cell chemotaxis, which are crucial for development of perivascular fibrosis. Therefore, using the flow cytometry, we evaluated perivascular inflammation, and we noticed that infiltration of MIR214 leukocyte into MIR214 animals were abolished, especially while induction of T cells, macrophages, and dendritic cells in wild-type T cell was observed. The knockout animals were protected against those changes. Additionally, also recruitment of T cells toward perivascular adipose tissue was 5 times lower after knockout transfer than wild-type T cell transfer observed in RAG1 knockout experiments. Therefore, we next studied the role of MIR214 in T cell chemotaxis in vitro using Boyden chambers and in vivo using an air pouch model toward chemotaxis which are involved in hypertension. And both approaches showed reduced chemotaxis of MIR214 deficient T cells in comparison to wild-type T cells. So this process was associated with the induction of relevant chemokines receptors, induction observed only in wild-type animals upon Ang2 infusion. So finally, Ang2 infusion caused a significant elevation of mRNA expression in protein level of many pro-inflammatory and pro-fibrotic factors, including TNF, interferon, IL-17, and IL-9, only in wild-type T cells, while MIR214 knockout animals were protected against those changes. And this can provide the potential mechanism how T cell MIR214 may modulate the perivascular inflammation and fibrosis. So in summary, we believe that hypertension affects MIR214 expression, which leads to the activation and transcriptomic changes in T cells, and consequently enhanced effect of cytokines production and expression of chemokine receptors, which intensify the migration to our vasculature, leading to inflammation and fibrosis, and consequently to arterial stiffness and accelerated vascular aging. So at the end, I would like to thank to all people and for their help and engagement during this study, and audience for your attention. Thank you very much.
Thank you, and I think it's a good time to start our discussion. There is a number of questions already on from our participants, and maybe Christian, I will start by asking to Nick the question about the role of pericytes in general in sort of cardiac regeneration, and is it possible that if pericytes may become fibroblasts, is it reversible? Is it a two-way pathway? Is there any thought that you could share with us about that?
Sure. Thank you, Tom. So there is a lot of literature on the multiple potential paths, differentiation paths that pericytes can take. And they have been suggested to be involved in regeneration. As everybody knows here, the field of cardiac regeneration has been marred by controversy, and there is no robust evidence that actually there are cellular processes, endogenous cellular processes that remuscularize the heart in a meaningful manner after infarction. There was, however, evidence with pericyte populations being administered in infarcted hearts. Paolo Madedu had done a lot of this work, suggesting that there may be a regenerative response that is induced. However, the problem here is the level of documentation, and tracking pericytes in infarcted hearts using lineage tracing strategies with in our work does not give any evidence of conversion into cardiomyocytes. Whether the cells have sufficient plasticity to produce cardiomyocytes, I would find somewhat implausible, because I think there are multiple factors needed to remuscularize the heart. And if there is a role for pericytes, it would be in repair by contributing to angiogenesis more than anything else. So any reparative effects that one may see may be due to an angiogenic stimulation rather than a conversion into cardiomyocytes. And the second question, it was the reversibility of the conversion. I think the pericyte to fibroblast conversion is not a major event in myocardial infarction, at least. And in some of the other pathologies that we looked at, is not the common event. That does not exclude the contribution of the pericytes in fibrosis, because of the potential of these pericyte-derived fibroblasts and the activated pericytes in producing growth factors as well. So there could be an effect that is paracrine in terms of activating the fibroblasts. Whether in general a fibrotic response can be reversed. This is something that has not been settled, because in experimental models, it is very difficult to study sequentially a model in which there is fibrosis, and then one can follow a process, perhaps an unloading process, a reduced mechanical stress process in which there is resolution of fibrosis. The cell biology and common sense and the logic suggests that more cellular and associative data as well suggests that more cellular fibrosis, meaning interstitial remodeling that is rich in cells and does not have large amounts of cross-linked matrix, should be easier to resolve than the established fibrosis that includes cross-linked matrix and would be resistant even to protease, to the effects of proteases. However, I think that's a field where there is a great opportunity to produce new insights, as even in a context where a resolution is feasible in a fibrotic lesion that may not be cross-linked, we do not understand whether there are conversions of fibroblasts, macrophages, perhaps vascular cells, which are important to resolve fibrosis, and how you get the degradation of the existing matrix without inducing inflammation. Anytime that you have degradation of matrix following injury, you have formation of fragments which are pro-inflammatory. Yet, if you have successful resolution, clearly that does not activate an inflammatory process. So how does the cellular environment in a resolving fibrous lesion achieve that is something that we do not know. The models are feasible, and this is a direction that one could pursue. Just interpretation is much more difficult, because you need somehow to be able to sequentially study the fibrotic response and then the resolution, which is not trivial when you think about the design of the study.
I agree. It's the question of resolution and reversion of fibrosis is something that I think we will learn in the next years. Christian, would you like to continue? And then
basically also following up on that. I mean, there's also a known role of parasite CCL2 and MIF in guiding myeloid cell recruitment into the perivascular space, and I was just wondering whether that actually gets differs in the fibroblasts after transition. So is that function lost, or is that preserved?
So what we know about the effects, the changes that occur in pericytes that converted into fibroblasts is that their expression of matrix genes is increased, their expression of growth factors is increased, their expression of integrins is increased. Expression of inflammatory genes is actually lower than in the pericytes which have not converted, which may suggest that any contribution to inflammation may be limited to the pericytes which do not convert. So there may be a dichotomy there. And what determines which subset actually converts, if it is a cluster that is prone to such conversion, and which cluster remains into the pericyte tract and into the mural cell tract, is not something we understand. A challenge with this study so far has been the problems in getting sufficient amounts of material from relatively small populations of cells, especially when you're sorting these populations from the infarct, the converted versus the non-converted versus the fibroblasts from other sources. So I cannot speak about the whole transcriptome study on these cells yet.
And linking to this question, what about fibrocytes? You know, there are studies studies, lineage tracing studies, especially in relation to perivascular fibrosis that is also present in the heart, but also peripherally, that's quite a substantial percent of profibrotic cells are actually originating from fibrocytes. What do you think about this contribution of this component?
Sure. So first, the definitions, right? Because fibrocytes, the definition that I would follow is the cells that come from bone marrow origin,
yes.
From circulating cells, because people have used the term in complex words with different meanings. So to make sure that everybody is on the same page on this. So the contribution of blood-derived cells is something that has been suggested for many years. And actually, the methodology was sound in terms of suggesting that by using chimeric animals, in some cases with lineage tracing as well, suggesting that macrophages may convert into fibroblasts. What speaks against it in the models that we most often used, which are infarction, pressure overload, is the studies, and it's not just one, but it's like from Jeff Molkentin, from Sylvia Evans, from Reza Erdehali. All three of them failed to find significant contributions of circulating cells. And perhaps a factor in the conflict there is how you identify the fibroblasts. For a cell that you don't really know what it is, it's difficult to study its origins. So you have to be very clear in what strategy you use to document the conversion. And this differs between studies. So, some studies have used non-specific markers, which like FSP1, which is expressed in immune cells as well, and in the macrophages and lymphocytes in particular. But even taking out this methodological aspect. So in conclusion to this, I would think that on the basis of what we know, I would consider the contribution of circulating cells to the fibroblast population rather small, rather limited, being informed by the majority of the robust studies using lineage tracing approaches with strong strategies to identify the fibroblasts. With one issue here, so I appreciate the differences in context. So you can have a situation in which there is an environment so rich in inflammatory mediators, such as a primary inflammatory condition, such as myocarditis, for example, in which you could have recruitment of leukocytes and of a diverse type of leukocytes in an environment which gives them immune stimulation, and perhaps at such an environment there could be possibly a conversion, but has not been documented. So I'm not a person that excludes anything. I just go by what the data show, and in a probabilistic manner in terms of suggesting what is the level of probability that a specific paradigm is true. So I would say at this stage, with the evidence that is out there, that the probability that immune cells contribute to fibrosis in models of mechanical stress or infarctive injury is very low on the basis of the totality of the evidence, but cannot exclude such a contribution in an environment where it has been less studied, such as primary inflammatory condition with a chemokine-rich environment, for example.
And in the studies of Alex, I wonder if have you looked at some of these animals? I think it also feeds into the questions of some of our viewers who are asking about the various sort of myogenic markers in some of these cells. Did you have a chance to look at this? And then maybe Pasquale can ask.
Myogenic markers in fibroblasts, like smooth muscle actin,
yes.
Or what do you mean?
So this is one question from one of the participants. The other question is did you look at the origins of these cells in your cells?
Yeah. So we can develop some inferences based on various algorithms to look, you probably seen some of them, velocity, monocle, etc. But I'm very tentative forming conclusions from these analyses. It depends on the parameters you use in these algorithms. I think to answer some of the comments Nick was making, I think we don't have the lineage tracing tools to answer some of these questions, and the precision in the lineage tracing tools are lacking. And some of the tools we're using were developed decades ago. And one of the things I would like to see happen more is using especially the single cell omics data sets to actually develop precise tools, for pericytes, for fibroblasts. I mean, there are certainly some markers that emerge now, which we can statistically show are very, very specific for fibroblasts in the heart, but not in the aorta, for instance. So
thanks.
We have looked at it
and the sort of smooth muscle actin positive that Guangping Li is asking.
Yeah, so I mean that was part of my point. We don't see smooth muscle actin positive fibroblasts in this model.
I missed this. Sorry.
But we do see smooth muscle actin, smooth muscle cells. So this is why I think it's important. I think this kind of challenges our view a little bit, at least my view of the primary drivers of fibrosis. I get the sense from this analysis, and this was a surprising finding, that myofibroblasts aren't the central driver that perhaps we thought it was. So I think in that sense it's a little bit it's instructive.
Pasquale?
I have one question, a technical question for Alex. Single cell sequencing cardiomyocyte is complex because they are large or multinuclei. So you are the expert. So can you tell me where we are in this respect in order to understand better?
So that can be accomplished by a number of means. I mean, you can do that by sequencing where cardiomyocytes are deposited into wells, or you can extract the nuclei of cardiomyocytes. And that's what we did for the analysis I showed you to consider cardiomyocytes. I mean, there's a compromise there. You lose RNA, of course, because you're only considering the nuclei of the cells, and you might lose consideration of multinucleation or ploidy, etc. So using single nuclear RNA-seq, it's very doable and relatively straightforward. So yeah,
okay,
that's one way of overcoming the size issue.
Perhaps also some question to Richard on your talk. So there are several questions. On the targets of microRNA 214, there's one proposition of SMAD7, but maybe you have also other ideas of targets that affect the T cell function in your model.
Okay, so I already replied about this. So, but this is SMAD7 was our initial target because it's linked with the protects against fibrosis. However, we did not notice any significant changes on the protein level in our model. So we very quickly give up. And in context to the mere expression myocardiocytes, so to be honest, I don't know what will be the difference between the fibrotic and non-fibrotic myocardiocytes. However, we see the difference that MIR214 is induced in hypertensive animals, and particularly in the left chamber, which undergo the profibrotic and pro-hypertrophic changes.
Also, probably relating to this, or inherent in this question, is whether it's a consequence or related also to the immune cell response that you see, or whether it's independent and simply occurring in the
And this I cannot try to challenge Alex a little bit by who is trying to say it's really not immune in females or so. I think, I wonder, can you elaborate a little bit or open a little bit towards this concept that the sort of immune and inflammatory regulation of this process is much more complex than just the fact if we see at certain time point an induction of inflammatory cytokines when you harvest the animals.
Yeah. So this was a surprising finding for us, right? I mean, we were essentially trying to validate or detect things people have been describing for so long. And indeed, we saw inflammation in the male hearts, but we often don't consider the female hearts. So that was kind of a feature we were trying to build into our study. We could see so many sexual dimorphisms, actually. I didn't get to time to talk about it. Just the fibrosis deposition, the distribution was different between females and males. But the inflammation aspect, and once again, it is surprising to see inflammation downregulated in the local cellular environment. I mean, these are agnostic methodologies we're using to detect these, and particularly endothelium, macrophages, and fibroblasts are downregulating circuitry and genes driving inflammation. These are the CCL ligands, for example. That's in both sexes. But in the male,
it's possible that it's temporally dependent. That it depends.
Yeah, absolutely, absolutely. That's critical, I think. I mean, for instance, in diabetes, I showed the high-fat diet context. At the time point we looked at, there was no fibrosis, but extensive inflammation. In the DB mouse model, we see inflammation, no fibrosis as well. But I know Nick's group has published 6 months up in a DB old mouse, there's fibrosis, and I think it's really important when we look at fibrosis. Or try to look at the DB mouse, for example, it's early on, and there's inflammation but no fibrosis. So I think the temporal aspects is really important. So we looked at one snapshot in time, but I guess the point is you can see fibrosis, you can see dysfunction of the heart, but no inflammation, suggesting to me at least inflammation is not a critical mediator of fibrosis, at least in this model, because the females don't show any inflammation. So that was a surprising finding, Tom. And I think what's happening is certainly at later time points, you perhaps see inflammation, but I think my intuition or sense there is that's probably an injury response because of the fibers and the tensile, the changing of the environment. For example, these activated fibroblasts, they express genes like tendomodulin, which is, which is present in the Achilles tendon, So I think there's going to be structural damage there, which then might elicit an immune response. So anyway, they're the insights we got from that study.
Thank you.
And what about Ng2 nestin positive versus negative subsets on the parasite and nestin expression in the fibroblast? That's also a question to both of you, Alex and Nick.
Yeah, we have not looked at nestin expression. I know Angelo asked that question in the chat, and there is a paper by Alex Pinto that actually he was using antibodies to nestin to identify these two different subtypes. We did not. I'm sure there are many ways to identify subtypes. Perhaps, from a transcriptomic viewpoint, the clustering algorithms are one way to go about it, and then you can have selected markers for flow cytometry or immunohistochemistry. I think these make sense when there is a distinct functional phenotype that you can relate to these. And so far for the pericytes, outside that dichotomy of the ones that convert to fibroblasts and the ones that do not, the majority that seem to be mural, we have not identified any other type of distinction, but we haven't done single cell work on this. Alex, if you have some single cell.
Yeah, look, nestin definitely, I can't remember it coming up on our radar. So, we haven't looked. In fact, I'm sneakily looking at my data set as we speak to see if it comes up. But no, look, the short answer is we haven't looked. What was the other marker, Christian, you noted?
Nestin, I think he mentioned.
Oh, yeah.
Yeah. That's basically that's related.
Oh, the NG2, on the other hand, I think it's at least using single cell transcriptomics, it is fairly restricted to pericytes. It's CSPG4 or something, I think, the gene. So it might also be expressed in a small subset of smooth muscle cells, but it's fairly restricted. But even then, I think the Cres, I think Nick's going about it the right way. We're doing the flora and validating the Cres restricted to pericytes. A lot of the markers that we've used in the past, lineage reporters, etc., when we use Cres and fluorescent reporters, we find actually that fluorescent reporter gene is in cells we didn't expect, so at least when we look at it by single cell transcriptomics. So I think it's a good opportunity actually to validate a lot of our Cre tools, and do the linear reporting experiments. But NG2 looks like a good one to me.
Absolutely, and that's for the inducible NG2 CRE because during development it's expressed by a large fraction of cardiomyocytes as well. So when we use the germline CRE, we had a lot of cardiomyocyte labeling. It's the inducible though that will target specifically pericytes, and there is a continuum in mural cells. So from vascular smooth muscle cells to pericytes to cells in between, depending on the hierarchy of the vessel. So you will get in arterioles number of cells target. You will get even in aorta about 40–50% of the vascular smooth muscle cells. There is not a single specific marker that will differentiate at this stage.
Maybe one more question to Ryszard. I find that quite striking, the effect of hypertension on the transcriptional control of microRNA 214 in the T cells. Is there anything known about the mechanism? Does that require Ang2, or is that independent mechanism? How does this work?
So I think it is independent mechanism because we also showed this that simple anti-CD3 treatment, which activates T cells, leads to the increase of miR2-4 level. And I think that Ang2 infusion and maybe the environment associated with hypertension can additionally induce this miR2-4.
Okay, but where's the microRNA 214 position? Is there anything known about the transcriptional regulation of that locus, or?
So generally, we try to understand how it could control this activation on migration and affect this fibrosis. However, it is known that microRNA 214 has more than 200 targets. And we found later after we tried different genes like ICOS, PTEN, STAT5B, but none of them were significantly downregulated and change in the knockout animals. And later we know that, for example, hydrazine treatment, which reduced the inflammation, fibrosis, and activation of T cells, also reduce the miR2-4 level. So after the hydrazine treatment, we don't see induction of miR2-4 in the T cells. But however, the single target could be very difficult, as we show on our heat map.
Yeah, Tom, shall we?
Do you want to summarize?
Or
yeah, I think if any question was not addressed, then we will ask our speakers to address, and our office will help to mediate this. Thank you very much to everyone for joining us here today, and maybe Christian can wrap up. I would like to thank particularly Nick, Alex, and Ryszard, but emphasize that Nick is really very associated and linked to cardiovascular research. He's one of our top reviewers, and I always appreciate the depth with which he really treats the papers, and with huge respect and extremely valuable commentaries. So thank you, Nick, for that, as well as for coming here today. Thank you to Alex and Ryszard. Christian, if you can just say a few final words.
Yeah, thank you very much, everyone. I think this was a wonderful example again for this format, how this can transition from basically the established experts to the young uprising stars in the field. I think it's wonderful, and I think we should keep it up. And I also noted, I think this was very nice this time, how actively you already basically addressed the questions in the toolbox here. So I think this is wonderful. If there's anything remaining, just basically continue to do that via email. Thank you very much, and we look forward to the next occasions on our CVR Discovery webinar.
We will come back after the summer break. Thank you very much.
Bye, Tom. Good night.
Thank you, Nick.
Bye, Christian.
Bye. Thanks a lot.
Bye.
Highly appreciated. Thank you,
Thank you.
Ryszard.
Have a
Thank
good
you,
day.
Alex. It was great.
No problems. Yeah.
Bye-bye.
Bye-bye.